Dear students, a very good morning to all of you. As you people are aware that in our last bio class, I started with the chapter cell division and I began with the introduction there where you people studied the structure of a chromosome along with the structures of nucleosomes, nucleotides, etc. And I believe that you people still remember that. If not, I request you people to kindly go through the book once again, and if you have any query regarding that matter, you can ask me on my number. As of now, I am beginning this session with the topic cell division. Formally, when you define cell division, it refers to the division of a parent cell into two daughter cells or maybe more daughter cells. So whenever a cell divides, that's a cyclic form, and you call it as cell division. When we study biology, cell division includes two different types. One is mitosis and the other one is meiosis. There is a very particular difference between the two cell divisions. The first one, if you can see, I have written here equational division, and the other side I have written reductional division. So the definition of mitosis and meiosis is actually hidden in this term only. When you are talking about equational division, you are referring to the maintenance of chromosome number whenever a cell divides. I mean to say that during mitosis, when a parent cell divides into two daughter cells, two offsprings, they are identical to each other, they are identical to the parent, and the chromosome number is also similar to that of the parent. That means the chromosome number is neither doubled nor halved. That's why you call mitosis as equational division. The other side, if you can see, meiosis, though in your syllabus meiosis is not included in detail, but whenever you are comparing the two, you should be having an idea what does meiosis means. I have written here reductional division. Reductional division refers to the chromosome number that is reduced to half. Whenever a cell divides into two, the offsprings receive only half the number of chromosome which is present in the parent cell. So I hope now by this time you understand what do you mean by equational division and reductional division. In mitosis, the chromosome number is maintained with the parent cell, and in meiosis, the chromosome number is reduced to half. I have written here a few more points where you can understand that for what purposes mitosis is carried out in the body and for what purpose meiosis is carried out in the body. Mitosis is done whenever your body cell is divided for growth purpose. That means the increase in size. If you people remember that an infant, a newborn, is smaller in size, and in due course of time the size increases. That means when you grow adult, your size, your body weight, everything is increased. That means the number of cells must have increased, and this is due to mitosis. The second one is repair. Repair means whenever you get injured, sometimes your skin is torn or your flesh is torn, or sometimes there is a bone injury, whatever. Whenever there is a kind of injury leading to the damage of cells, in that case, mitosis is the process which includes the repair. It helps in repairing of damaged cells by making their own kind, more of their own kind. And the last one is a replacement. Replacement means in our body cells, whenever we go with the lifespan of the cells, every cell has its own lifespan. That means if you talk about an RBC, there is a red blood cell. It survives for 120 days. If you talk about WBCs, these are white blood cells. They survive for moreover 2 weeks. But after that, they die naturally. What is the thing that replaces this loss? It's mitosis, dear. Mitosis helps in replacing the lost cells. But when you talk about, on the contrary, when you talk about meiosis, meiosis is included whenever a body goes for reproduction. That means particularly you have to specify that it is involved in formation of gametes. Now, what are gametes? Gamete is the other technical name for the reproductive cells produced in your body which participate in sexual reproduction. Always remember that gametes receive half the number of parental cell of half the number of chromosomes from the parental cell, and this is just to maintain the normal number. Whenever a male gamete fuses with the female gamete, the zygote, the outcome that is formed after fertilization, should form a normal number of chromosomes. I hope you people understand by this time that cell division is the division of parental cell into 2 or more daughter cells. It includes mitosis and meiosis. Mitosis is equational. Maintenance of chromosome number is involved here. Meiosis is reductional. That means the chromosome numbers are reduced to half. I'm proceeding further. In your syllabus, you do have the difference between mitosis and meiosis. And this is a tabular form where you can see that in one column mitosis is written, the other one is meiosis, and on the leftmost column you can see the fields are given. I mean those headings on the basis of which you are going to compare the two. The first one is the definition, and it repeats that mitosis maintains the number of chromosomes, meiosis reduces the number of chromosomes to half. The other field is where does mitosis occurs. That means mitosis includes, it is occurring in all organisms. There is no specificity about the cell. But when you talk about meiosis, reproductive cells are the content. That means meiosis occurs only in reproductive cells, so that these reproductive cells which are involved in sexual reproduction, these reproductive cells receive half the number of chromosomes which become normal after fertilization. The third heading is type of reproduction. That means mitosis occurs in us too, but this is the process which allows asexual reproduction where a parental body divides into 2 identical offsprings. The way you see in amoeba. Amoeba divides by binary fission. That's an asexual mode of reproduction, but generally you can call it mitosis only because one parental cell divides into 2 identical offsprings. But when you talk about meiosis, it is specified that meiosis occurs only in those organisms which undergo sexual reproduction. The next field is genetics. The offsprings which are formed, are they identical to each other or they are different? So mitosis allows the production of identical offsprings, whereas meiosis allows the production of different offsprings. There is a reason behind. If you can see that next 2 headings are crossing over and homologous chromosome, pairing of homologous chromosome. You should understand that these 2 headings are involved in, they are very particular events in meiosis. Whenever you are going to study meiosis in detail, you will come to know that there is a phase, prophase 1, where crossing over and homologous chromosome pairing takes place, which is not present in mitosis. So these 2 are very important differentiations. No crossing over in mitosis, crossing over is there in meiosis, no pairing of homologous chromosomes, and pairing of homologous chromosomes. Then you have the number of divisions. This is again an important point because in mitosis only one cell cycle is there. That means you include prophase, metaphase, anaphase and telophase. But when you talk about meiosis, it is completed in 2 sub-cycles, that is meiosis 1 and meiosis 2. The last heading is number of daughter cells produced. This is a very common difference. In mitosis, always 2 diploid cells, diploid because the chromosome number is maintained. So if a diploid parent is dividing into 2, it is going to produce 2 diploid cells only. But when you talk about meiosis, 4 haploid cells, that means the chromosome number is reduced to half. 4 haploid cells are produced. This is something you need to learn. So I hope that you go through this tabular form very seriously. I'm moving further. Please see this page again very seriously with great focus. I have written the heading phases of mitosis, but I have started with interphase. Most of the students, they tend to ask me during the class that is interphase one of the phases of mitosis? And my answer is no, not at all. Interphase is just a name for a resting or preparation phase of a cell where a cell is not going to divide. It is just undergoing certain changes. And this resting phase is divided into gap 1. You call it G1. Gap 1 phase, synthesis phase, S stands for synthesis phase, and gap 2, this is G2. Gap 1 includes protein synthesis, the cell grows and its cytoplasm is also doubled. Gap 2 is also similar to G1. That means whatever event are going on in gap 1, they are going to be carried out in gap 2 too. But synthesis phase is slightly different because this phase includes the synthesis and replication. That means the DNA is synthesized here and also the DNA content is doubled here. So increase in DNA content is also the point of consideration. Otherwise, normally, whenever the question is what are the 4 phases of mitosis, you say phase 1, prophase, phase 2, metaphase, phase 3, anaphase, phase 4, telophase. If you can see, I have written here early and late. This is just to understand that when prophase begins, it is early. When it is about to end, it is late. Similarly, anaphase, when it begins, it is early. When about to end, it is late anaphase. Out of these 4 phases, I have mentioned here specifically because this is another important question in boards, that which is the longest phase of mitotic cell division. In that case, must understand prophase is the longest phase and anaphase is the shortest phase of cell division. After these phases, there are 2 semi-technical terms here, if you can see at the bottom, karyokinesis and cytokinesis. In nutshell, I have written division of nucleus, and for cytokinesis, I have written division of cytoplasm. A very simple idea to learn this. Karyon word means nucleus and kinesis means break or division. So division of nucleus is karyokinesis and division of cytoplasm is cytokinesis. Understand that karyokinesis starts when the last phase, that is telophase, is about to end. Please go through this page seriously. This is very, very important with respect to boards. I'm moving further. We are just now going to understand the main events that take place during different phases of mitosis. The first phase is prophase. I have written here early and late, and for your easiness, I have divided the whole screen into 2. Early prophase followed by late prophase. This is the beginning of prophase. This is the end of prophase. It is a labeled diagram, so you can see I have written the specific features of both the phases in detail. So we just let us go through these events. Centrosome splits into centriole. So you see that one centrosome was earlier present and now it is divided into 2. Once it is divided, you start calling it centriole. Spindle formation begins. That means, if you can see, there are small microtubules which are joined together to form spindle, and this is just the beginning. They are not formed completely. The third one is chromosome begin to shorten and thicken. That means as of now, till now the chromosomes were thread-like, right, interwoven, but now they supercoil over themselves to be shortened and thickened. That means you may call it condensing. If you can see, the label says chromosomes start to condense, become short and thick. And the last one is in early prophase, nucleolus disappears. I hope you people understand that nucleolus is a small portion present within the nucleus and it is responsible for formation of RNAs, that is ribonucleic acid. On the other side, if you can see, this is late prophase. Yeah. Some people do call it pro-metaphase because this is prior to metaphase, right? And the changes here are spindle formation is completed. In early, it started. Now, in late prophase, it is completed. If you can see, all the spindle fibers are connecting the 2 poles. Chromosomal condensation is also completed. That means now finally the chromosomes are condensed, supercoiled, thickened. They are very distinct. They are very easily visible under the microscope now. And the last one is nuclear envelope disappears. That means whatever nuclear membrane you were having here, it starts dissolving in late prophase. Please go through these diagrams, the status of chromosome and the events very seriously. Another thing that I would like to add here is that sometimes your question says, draw prophase with 2 pairs of chromosome or draw prophase with 4 chromosomes. That means this is the diagram with 4 chromosomes. I'm moving further. Followed by is the phase metaphase, and this is the easiest one to be explained because here only one event, rest of the events have already taken place in prophase. This is the phase where only one event takes place, but that's the most important one. There is an equatorial plate laid down in the center. It is an invisible, assumed plate. There is no such structure. It's just an equatorial plate which is assumed on the basis of settlement of chromosomes in the center. How these chromosomes are arranged in the center? The centromere of the chromosome attaches to the spindle. Every chromosome is attached to the spindle along its centromere. This is the particular feature of metaphase. Proceeding further. Anaphase, the shortest phase I should say, early and late. That means when it begins, it is early. When it is about to get over, it is late. As of now, the diagram that you can see is early metaphase, right? Let us discuss what are the features here. This phase is characterized by splitting of chromosomes. That means during the previous phase, metaphase, the chromosomes which were arranged on the equatorial plate, now they split from each other. That means this is the phase, the splitting actually makes the chromosome number doubled. The splitting of chromosomes take place and chromosomes start moving towards the pole. So if you can see in previous phase, metaphase, the chromosomes were settled here, but now they are split from each other and half of the chromosomes are going to one side, half of the chromosomes are going to the other pole. And this movement is actually supported by the pushing or pulling action of microtubules. That means the spindle fiber starts contracting, and just because of this, the chromosomes, they are pulled towards the respective poles. Late prophase, that means in early anaphase, the chromosomes are just, they have just begun to move, and in late prophase, these chromosomes would be near the equatorial, near the poles. I have written it here, chromosomes near the respective poles. The important feature here is spindle begins to reduce. That means whatever spindle fibers were formed earlier, now they will start getting dissolved. And if you can see in late prophase, I have written a slight constriction or furrow in the cell membrane arises. If you can see, the diagram does contain a slight constriction in the center. A furrow is formed here. Now this is the point where this cell is going to divide in future. Moving further, telophase. Actually the second last phase, because this phase, if you can see the diagram, now constriction is more narrower, right? And you can see particular features in the diagram. The first one is spindle completely disappeared. You cannot see any more spindle there. Nucleus and nucleolus, which were present there in the beginning of the cell cycle, they reappear. So the nuclear membrane would start reappearing and the nucleolus would also reappear. If you can see now, this nuclear membrane would join together to form a complete nucleus. That means in the beginning you started with one nucleus. Now you are ending up with two. You name the term as karyokinesis. That means division of nucleus. But as of now, if you can see, there is no division of the cytoplasm. You do have a place here. You are labeling it as a furrow, but it is still not divided. So for this division, we have another phase. You call it, it's not a phase, it's just a step rather, cytokinesis. Cyto means cytoplasm. Kinesis means division. So division of cytoplasm. In animal cells, the division occurs at the place of constriction. Wherever you had that furrow, this furrow gets more deeper and deeper, and ultimately this portion is cleaved off, dividing the whole cell into two daughter cells. But when you talk about plant cell here, In plant cell, there is no furrow formation. There is a straightaway cell plate laid down at the center, and from along this cell plate, the whole cell divides. So must understand this, that in plant cell, there is no furrow formation. A cell plate is laid down in the center, and this cell plate allows the division of plant cell into two. Another important thing is, sometimes when you have the question in boards, in boards any of the phase can be given in the diagrammatic form and you are asked to identify, or you may be asked to write the particular features of them. You may be asked to write which one is longest, which one is shortest, and sometimes you may be asked that identify whether the cell is a plant cell or animal cell. Straight away look to two things. If there is a cell plate labeled above, you say it is a plant cell, or otherwise if you can see a double membrane, that means the outer one is cell wall and the other one is inner one is cell membrane. Whenever you find these two labeling, cell wall and cell membrane, your cell is plant cell. This is how you identify whether the cell is animal or plant cell, and this is how mitosis is completed. Once these cells are divided into two, now this individual cell would be performing another mitosis independently. Here your topic is ended, and I believe that I have made myself very clear about cell division. If you have any queries, you can put your queries up on my personal number and I would be answering all your queries on group. I wish you all the best. Thank you.